Class 10 Biology, Chapter 3, How do organisms reproduce? Video number 2. Topics covered in this video are features of asexual reproduction and types of asexual reproduction. Under this, I'll be discussing about fission, fragmentation, regeneration, budding, spore formation and vegetative propagation. Asexual reproduction. It is a type of reproduction in which a single parent produces young ones without the involvement of formation and fusion of gametes. That is, in this process, there's only one parent involved and the process does not involve formation or fusion of gametes. So let's see what are the important features of this process. The first important feature of this process of reproduction is it is uniparental, that is the process involves a single parent, right? Whereas in case of asexual reproduction, about which you'll be learning in the next videos to come, that's biparental, that is there are two parents involved. The next important feature is there is no formation or fusion of gametes, that is gametes are not involved in this process and the process takes place in a shorter period of time, that is it's a faster method of propagation. That's another important feature. It's a rapid method of multiplication or propagation. And the young ones produced as a result of asexual reproduction are genetically identical to that of the parent. That means that the variations that are produced, they are quite less. Try to recall, in my previous video I had discussed what are the reasons for variations. In case of asexual reproduction, the only cause of variation will be error in DNA copying. So it's only because of the error that happens during DNA copying that results in variation. And try to recall, it was told that these errors should be minor error, because if there is a major error, then the resultant copy of DNA will not be able to work with the cell apparatus and the resultant cell will die. So if it's error in DNA copying being the only reason for variation in asexual reproduction, and that too the error should be minor error, this shows that the young ones that are produced as a result of asexual reproduction will be genetically identical. They'll be same as that of the parent with very slight variation. And because of very little variations being produced, the process is not so important for evolution and formation of species, because for evolution, for formation of species, variations they serve as raw materials, and here variations that are produced are quite less. So these are the features of the process of asexual reproduction. Now coming to the types of asexual reproduction. Here the slide shows the different types of asexual reproduction, namely fission, which occurs in unicellular organism, then we have got fragmentation, then you've got budding, regeneration, vegetative propagation and spore formation. In all these processes, there is only one parent involved, first important thing, and there is no involvement of gametes. So gametes are not involved. So let's study about each of this process one by one. First, let's know what is fission. Fission, the word fission means to split. It is to split. So it is a process of asexual reproduction in which the parent cell, because it occurs in unicellular organism, so the parent cells splits into two or more daughter cells. So that is what is fission, a type of asexual reproduction in which the parent cell splits into two or more daughter cells. So accordingly, whether the two daughter cells are formed or more than two are formed, fission is of two types. They are binary fission and multiple fission. So these are the two types of fission. Fission can either be binary fission or it may be multiple fission. And the best example of binary fission is the fission that occurs, or the reproduction which occurs in case of amoeba, paramecium. They are all examples, or leishmania, these are all examples of organisms that multiply by fission and binary fission. And remember that these unicellular organisms, they generally multiply or reproduce by this method when the conditions are favorable. Okay, but amoeba, it multiplies in a normal way also, it multiplies in this by this method, but then most of the time you find that when the conditions are favorable, those unicellular organisms, they multiply by this method. So let's see how fission occurs in amoeba. Amoeba, as you all know, is a unicellular organism. It's a protozoan which floats in water, and when it has to multiply, first amoeba will grow in size, that is, it will become larger in size, and once it has become larger in size, now the division starts. And as already said, that whenever reproduction occurs, the first thing that will happen is the nucleus or the genetic material that is contained in the nucleus will make a copy of itself. Means the division will start with the duplication of the genetic material. So here first the nuclear division occurs. The genetic material that is contained in the nucleus will make a copy of itself, and now the nucleus divides by elongating, as you can see in this diagram, right? So the nucleus elongates and starts dividing, and by the time the nucleus is divided into two, you also find a constriction, that is, a furrow develops in the cell, right? In the site in the cell membrane, you find there is a furrow developing, like here as you can see in this diagram. The nucleus is dividing, and by the time the nucleus divides into two, the cytoplasmic division also has to happen. So this happens by development of a constriction. This constriction keeps on deepening, and as it deepens, the constriction will finally totally divide the cell into two, and thus two daughter cells are formed. Not just amoeba, paramecium, leishmania, these are also, or the euglena, they also multiply by the process of binary fission. But you find there is a difference. In case of amoeba, you find that the amoeba has got no shape. It keeps on changing its shape. And so since it's not having an organized shape, you find that amoeba can divide in any plane. It can be this way, it can be longitudinal, so it can be dividing. It can be in any plane. Amoeba can divide in any plane. But other unicellular organisms like Leishmania. Leishmania is a protozoan which causes the disease Kala Azar. So Leishmania, it is an organism which has got a bit organized shape. It's single cell, but you find there is a flagella present on one side. And because of this, in case of Leishmania, you find that fission is always longitudinal. Okay, because the nucleus will divide, and as it is seen in this, and flagella, there will be two flagella made, right? And then finally the cell divides longitudinally. Whereas in case of amoeba, as I said you, fission can be in any plane, because amoeba is not having any organized shape. Okay, so here you find in amoeba it can be in any plane. One plane is shown, but then the fission can be in this plane also. It can be this way, that is transversely, so it can be in any plane. But in case of Leishmania, in case of euglena, you find fission is longitudinal, and here this diagram you find in case of paramecium, fission is occurring transversely. So those unicellular organisms which have a bit organized structure, you find that the division it occurs in a specific plane. One thing very important to note is that once the daughter cells are formed, once the cell has divided into two daughter cells, the parent's identity is lost. So parent will no more exist. So this is how fission occurs, binary fission occurs, where the parent cell divides into two daughter cells. And whenever division occurs, first it will be nuclear division taking place, which will be followed by the cytoplasmic division. Now let's see what is multiple fission. Multiple fission, it is a type of asexual reproduction in which the parent cell divides into it divides into more than two cells. So more number of daughter cells are formed. And the best example for multiple fission would be Plasmodium. Plasmodium is the malarial parasite which causes the disease malaria. So Plasmodium, it divides by this method. Now let's see how this process occurs. And here you find Plasmodium, it's a normal way of division in case of Plasmodium, but then in many of the other unicellular organisms which are multiplying by multiple fission, there you find they will divide during unfavorable condition. But in case of Plasmodium, under normal condition also, it multiplies by the same method. So here, as you can see in this diagram which is shown in the slide, you find first what happens is the nucleus which is present inside the cell of the parent cell will divide many a times. It will divide many a times. The nucleus divides many a times, forming many daughter cells inside. Like if this is a cell, there is a nucleus present. So first the nucleus will divide many a times and form many daughter nuclei. Once the daughter nuclei are formed, now each nuclei or nucleus will get surrounded by a bit of cytoplasm and will develop its own membrane. Thus, you find that inside the cell or the cells, there are many cells formed. Then the outer covering of the cell, if it is a parent cell, the outer covering or the cyst, if it is unfavorable condition, there will be cyst present. So the cyst will break and then many cells are released at the same time. So this type of division in which one single cell or the parent cell divides into many daughter cells, this is what is called as multiple fission. And both binary fission and multiple fission occurs only in unicellular organism. They will take place only in unicellular organisms, right? Now let's study about the next mode of reproduction, asexual reproduction, that is fragmentation. So fragmentation, it is a type of asexual reproduction in which the parent, after growing to a certain size, will divide into many pieces. Each piece is referred to as fragment. Okay, each piece is referred to as fragment. The fragment then undergoes repeated cell division and a complete individual is formed. So this fragmentation occurs in multicellular organisms. Multicellular organisms which have got a very simple body design, where you find that the cells are alike. Okay, so you don't find a well organized cell structure, body structure. So there in simple multicellular organisms like Spirogyra, the example that is given in your NCERT is Spirogyra, it multiplies by this method. So what do you find in this? Like in case of Spirogyra, Spirogyra is a green algae found in water. So as the algae filament, this is a filament of the algae, as it grows in size, the filament may break into pieces. Okay, so here you can see in this diagram, the filament has broken into 3 pieces. It can be 3 or more number of fragments. Then each fragment undergoes repeated cell division. So by repeated cell division, because there is no differentiation occurring, it's just cell dividing many a times, and finally a complete individual is formed. But then this fragmentation is possible only in simple multicellular organisms, organisms which have got very simple body design. Okay, they multiply by this method. The process of fragmentation is not there, or it's not possible on organisms which have got like us, or the other organisms which have got a complex body design. There you don't find that they divide by the process or multiply or reproduce by the process of fragmentation. And why is it not? Because in case of multicellular organisms with a higher body with a complex body design, the body is not a random collection of cell. You don't find your body, our body, if you take ourselves as an example, our body is not just a collection of cell. Rather you find that our body, it is made up of huge number of different types of cells, cells which organize and form tissues, organs and organ system. And each specialized cell, tissue, organs and organ system has got a specific function to perform, and they have got a specific location in the body. Hence, one cannot think of that the body, like how what happens in fragmentation. So in case of a multicellular organism like us having a complex body design, one cannot think of that the body dividing into pieces and then each piece developing into a complete organism just by simple cell division, because the body is organized, it's well differentiated. So this is not possible. It's not possible for a multicellular organism to divide by this method, because you've got different types of cells performing different function. Okay, and so in case of multicellular organism, because there are specialized cells or cells specialized to take up specific function, so reproduction in such organism will be a function of specific kind of cell which is capable of growing, a cell that is capable of growing, then proliferating, that is dividing into many cells, and then differentiating. So these cells that are formed should be capable of differentiating and then forming a complete individual. So this special kind of cells are present in such multicellular organisms. Like us, we have got the gametes which fuse and form the zygote, and it is the zygote that is formed will then divide, that is, it will proliferate and make up other kinds of cells which will finally develop into an individual. Or in case of other organisms, you've got those undifferentiated, unspecialized cells which are called as the stem cells. These stem cells are the unspecialized cells which have a capacity to proliferate, that is to multiply, and then they make other types of cells which will form a complete individual. So multicellular organisms with complex body design cannot multiply by simple fragmentation process, where the body is formed by just cell to cell division. There is cell division required, there is differentiation also required, and then the individual will be formed. So here you find that there are there's you find in case of multicellular organism they use a different mode where you find special kinds of cells are involved. Now let us go with the other method of propagation, which is regeneration. In fact, it's not considered as a type of reproduction. Why it is not considered, we'll discuss later. So first let us know what is regeneration. Regeneration is the ability of the body to get back the lost body part. If suppose the body gets cut accidentally, if the body gets cut, then the ability to get the lost body part is what is called as regeneration. And you find regeneration here. Two examples are shown over here. This is planaria and hydra, but you also find regeneration occurring in case of starfishes, where you find that if the arm of the starfish gets cut, then it has a capability of regenerating its lost arm. Okay, so in your NCERT book about planaria, it's mentioned that how planaria multiplies or propagates by the process of regeneration. So how it happens, let us see. Like if suppose planaria, which is a freshwater multicellular organism which is having an organized body. I just now had discussed about fragmentation, where I said that the body just divides into pieces, and then each piece by just cell to cell division develops into an individual. Here in this process of regeneration, it's not just simple cell division giving rise to the new individual from a piece. Now let's see how this happens. If suppose planaria accidentally gets cut into pieces, so here you can see there are 3 pieces formed. Now once it gets cut into pieces, then the cells, those unspecialized cells which I was talking about, which are present in the body, they will repeatedly divide. So the cell, suppose this is a if suppose this is one piece of planaria, how it's going to develop the complete body, because planaria and hydra, they have got a higher power of regeneration. They can get an entire individual from a piece. So here what happens is that those unspecialized cells which are present in the cut region, they will first proliferate, that is, they will first divide and form a mass of cell. So mass of cell is first formed, and then these cells will differentiate, take up different tasks, and finally from that mass of cell a complete individual is formed, as shown in this. So here it's because of those unspecialized cells present which will first proliferate, that is, they will multiply, then they will differentiate into different types of cell, and after differentiation the lost part is completely gained. So from each of the piece which has from each piece into which the planaria has got cut into, a complete individual is formed. And here it is not just cell division, continuous cell division, which is giving rise to an individual. It's cell division occurring and then that mass of cell that is formed, that differentiates, and after differentiation you find a complete individual form. So in case of planaria, this reproduction, it occurs in this way. Right. Now this you find in case of hydra also. In case of hydra, if accidentally hydra gets cut into pieces, then each piece of hydra, even if the tentacle gets cut, then from the tentacle you'll find first the cells, or those unspecialized cells, will multiply and a complete individual will be formed. So hydra also has got a high power of regenerating an entire individual from a small fragment of piece by cell division and differentiation. But this power of regeneration is quite limited in case of higher multicellular organisms like lizards. They can regenerate their lost tail, right? We have a power of regenerating which is highly reduced to just healing up of the wounds, right? In case of starfishes, they can get their lost arms. So this power of regeneration is very limited in higher organisms, because in case of higher organisms the regeneration is regulated by the hormonal system. So that's why you find it's quite limited. The power of regeneration is quite limited in higher multicellular organisms, but lower multicellular organisms, they like hydra and planaria, they have got a very high power of regeneration. Now the next thing is, as I said you in the beginning, that regeneration is generally not considered as a type of reproduction. Why? Because in case of regeneration, it's only by when an organism gets accidentally cut into pieces, then they will get back their lost body part. So it's a way of getting or regenerating the lost body part, and this will happen when the organism gets accidentally cut. And it's not that for reproducing the organism will be waiting for someone to cut it into pieces. No organism will wait for it to be cut into pieces for reproducing. It's only that if suppose an organism gets cut into pieces accidentally, if it's accidentally, okay, so if accidentally if it gets cut into pieces, then each piece is capable of regenerating the lost body part. Okay, so this is what is regeneration. The next mode of asexual reproduction is budding. Now, what is budding? It is a process in which an organism produces a small outgrowth. The organism produces a small outgrowth on its body surface. With this outgrowth is called as bud. The bud then develops into a complete individual, and once it has developed into a complete individual, it detaches from the parents and leads an independent life. It may also remain attached to the parent. In certain cases, the bud, after complete development, may remain attached to the parental body. But here what is more important is that the bud, it is formed on the surface, the bud develops, and then it forms a complete individual. So hydra multiplies by this method. So let's see how hydra multiplies. First, let's see how this happens. In case of hydra, you find that again, in case of hydra also, in budding also, you find those unspecialized cells which I had discussed about in regeneration. They are the ones who are active over here. So in those unspecialized cells which are present, those which are called as referred to as stem cells, present on the lower surface of the body, they repeatedly divide and then they form a mass of cell. This mass of cell is called as the bud. Okay, once the bud is formed, the differentiation will occur. The cells which are present in this bud, they will transform into different types of cells, and thus the bud will develop into an individual. Once the bud has developed into a complete individual, like in case of hydra, tentacles and other body parts are formed, the mouth is formed, the cavity inside is formed, and once the bud is totally formed, this bud, when it becomes nutritionally independent, means it can lead its own life, then this bud gets detached from the parent body and then it leads an independent life. Okay, so this is how hydra multiplies by the process of budding. And again, yeast is a unicellular fungi which also multiplies by the process of budding. If you take yeast in sugar solution and leave it for some time, you'll find that the yeast cells inside the sugar solution, because they get nutrients, so they start multiplying and they'll grow in number. A small drop of it, if you take under microscope and see, you'll find many buds attached to the parent cell. Right, so let's see how this process occurs. So in case of yeast, again as I said, your budding is a process by which the parent divides or asexually reproduces by producing an outgrowth which develops into a complete individual. Right, so in case of yeast, how it multiplies is that first on the parent cell wall a small bulge appears. This bulge that appears is called as a bud. Right, then the nucleus which is there inside will divide. The daughter nuclei will go into the bud and the parent will be retained, and parent nuclei will be retained in the parent cell. And finally, the cell wall between the parent cell and the bud will be laid. Many a times it happens that the bud may get detached, but in certain cases you will find that the bud, before getting detached to the parent, will develop its own bud, and in this way, under microscope when you observe, you'll find that there will be chain of cells or the buds present, a chain of yeast cells will be formed. So this you find in case. So it may be that the bud immediately may get detached, or before it gets detached, it will form its own bud, and so under microscope when you see, you will find a chain of cells. So yeast also multiplies by the process of bud. The next mode of asexual reproduction is spore formation. Spore formation is a type of asexual reproduction in which organism reproduces by producing tiny microscopic unicellular bodies called as the spores. Okay, they are called as the spores, and these spores, they are produced asexually. These spores generally they are covered by a hard protective covering which can help the spores to withstand unfavorable condition. Once the condition becomes favorable, the spore has a capacity to germinate and form a complete individual. So here rhizopus or the bread mold, it multiplies or it propagates by this method. If suppose you take a piece of bread, moisten it and keep it in a warm humid place, you'll find like after some days you'll find a cottony growth over the bread. Now you just take a lens, a magnifying lens can be taken, and when you observe, you'll find that entire over the bread, you'll find such thread-like structures present. These thread-like structures are referred to as hyphae, and these are the these hyphae which spreads over the bread. They don't produce any spores. when the conditions are favorable over the hyphae, these horizontal hyphae, they produce vertical stick-like structures which are called as sporangiophores. Okay, and on these sporangiophores, they bear at the tip, they bear blob or bulge on the tip, which are called as sporangium, and these sporangiums are the ones which produce spores. So once, like initially the sporangium, like when the sporangium is formed, inside the sporangium you will find there will be many huge number of spores produced. Each sporangium inside, you'll find there will be many spores produced asexually. Okay, and after the sporangium is mature, then you find that the sporangium will burst open, and the spores which are there inside, these spores, they are released out. Okay, each spore, it is unicellular. Each spore is just made up of one cell, and it is protected by a hard protective covering. So there is a hard protective covering present which protects the cell inside, okay, from all kinds of unfavorable condition. That's an advantage of spore formation. The spores that are produced, they have a hard protective covering which protects it from unfavorable condition. That's one advantage. And the spores, they are very minute, they are very small in size. You can see here they're very minute, very light, and as the spores are quite light, they get easily dispersed. So this will help in easy dispersal of the spores, and the hard protective covering will protect the spores or the cell inside from unfavorable condition. When the conditions are favorable, suppose the spores, when they spread in the air, they are present everywhere. So the moment the spores come to lie over some material which has got enough of moisture, they are nutrient present, the conditions are favorable, then the outer covering of the spore will crack open, the cell inside will germinate, and then finally again those thread-like structure or the hypha is formed, and the entire body of the fungus will be formed. So this is how the organism like rhizopus multiplies through spore formation. And I repeat, the advantage of spore formation is at a time huge number of spores are produced. Second, the spores are very small in size and they are very light. So as they are very light, they get easily dispersed. And the next advantage is that the hard covering which is present, the protective covering that is present over the spores, protects the spores from all kinds of unfavorable condition. Now, so far I have discussed about the methods of propagation, asexual reproduction in simple organisms. Okay, now let us study about the method of propagation that is seen in plants. Higher plants which are having a complex body design, they also multiply asexually, and they multiply asexually by the process of vegetative propagation. Now what is vegetative propagation? It is a type of asexual reproduction in which the parent plant, the plant produces new plants from its vegetative parts. And what can be the vegetative parts? The vegetative parts may be roots, it can be the stem, or it can be the leaf. Okay, flower is the reproductive part of the plant which takes part in sexual reproduction. So from the roots, stem and the leaves, also in certain plants you find new plants arise. Like in case of bryophyllum, this is bryophyllum. Bryophyllum, it is a succulent xerophytic plant, and what you find is in case of bryophyllum, when as the leaves grow older at the notches, as you can see in this, it's a in this picture of bryophyllum leaf, you find at the margin in the notches tiny buds appear. Okay, these are the buds. So once the leaf has become old, you find that the notches, the buds develop, the buds grow into small leaf plantlets. So these are the plantlets that develop. Once the plantlets have developed roots and all the things, they grow in size, then they fall. They will fall from the leaf, and once they fall on the soil, these buds will again grow in size and develop into a complete individual. So here you find in case of bryophyllum, new plants are arising from the leaf. Whereas in case of potato tuber, which you all must have seen, from the tuber of potato, that is a modified stem. In case of potato, in case of ginger, you find it is from the stem, that is there are buds present in the stem, and through those buds you find a complete plant emerging out. So in case of potato, you find it is through the stem. In case of sweet potato, propagation will be through root. You've got onion and garlic where you again find propagation occurring through the stem. Okay, so these are all what examples I have shown you. They are the methods of natural vegetative propagation. Vegetative propagation is also artificially done through human effort, and those different methods of artificial vegetative propagation includes cutting. So here what is shown in this slide is cutting, right? Then roses, they are propagated by this method. Then you've got another technique which is called as layering. As you can see here, the lower some of the shrubs like the lemon, hibiscus, jasmine, they can be propagated by this method. The lowest branch of the shrub is bent, and a portion which is to be placed under the soil. From there the outer bark will be removed, then the branch is bent, and then that portion will be covered with soil, and some heavy material like stone will be placed over it. You just leave this setup, this entire plant undisturbed for some time, keep on regularly watering it. After some time, the part that was covered in the soil, that part will develop roots. Then from here the plant or the branch can be cut, and now you've got a complete plant. So this is what is called as layering, right? Then there's another method which is called as grafting. So these are the different methods of artificial vegetative propagation. Now there's another way the plants can be propagated artificially, and that is called as tissue culture, where in this method, let's see what happens. First, a small portion in tissue culture, it is actually a technique in which a small tissue of the plant body is grown in a synthetic medium. Okay, it's grown in a synthetic medium, and then you get a complete plant out of it. So this is done in case of tissue culture. So first, in tissue culture, from the tip of any plant like roses or the orchids, you find this being done, or banana, they are propagated by this method. So from the growing region, as you can see here, you know that meristematic regions are present at the tip, the apical meristem. So from that, a small portion of the tissue is taken out as shown over here. This is called as the explant. Now, the explant will be properly sterilized, and then in a test tube, the explant is put over a nutrient medium. The nutrient medium will have various plant nutrients. There will be hormones like auxins, cytokinins in various concentrations kept, and this entire procedure is done in aseptic condition, sterilized condition. So after some days you find that this explant, the tissue that was put, will grow in size by repeated cell division and form a mass of cell which is called as the callus. Now the callus, it will be cut into tiny pieces, and after that the callus is again put in different nutrient medium. After some days you will find that the shoot differentiation, first it's a cell division forming a mass of cell, then differentiation will occur, the roots will emerge out, leaves or shoot will emerge out, and once a complete plantlet is formed, this plantlet is shifted into small pots, okay, and then it is then sold in the market. So this technique of propagating plant from a small tissue taken from the parent plant and then being grown in a nutrient medium is called as tissue culture, or it is also called as micropropagation. It's also called as micropropagation. So these are the different methods of asexual reproduction. Coming to what are the advantages of vegetative propagation, one should know what are the advantages. The first advantage of vegetative propagation is that it is a rapid, cheap and easy method of multiplication, right? A faster method of multiplication, because here the plants will produce flowers and fruits much earlier than those plants which are propagated through the seeds. The young plants that are produced by vegetative propagation are genetically identical to the parent plant. Okay, that is another advantage. And there are many plants like you've got jasmine, okay, rose, banana. These plants, they do not produce seeds, or in certain cases even if seeds are produced, the seeds are not viable. So in such plants, vegetative propagation becomes the only method of propagation. So that's about the different methods of asexual reproduction. Now on the basis of my previous video, the first video that I had sent, and this what you have learned today, here are some questions that you have to answer. Let's see what are the questions. First is, what is the importance of DNA copying? The second question is, what is the importance of variation produced during DNA copying or DNA replication? Differentiate between asexual and sexual reproduction. Why are the young ones of asexually reproducing organisms genetically identical to that of their parents? How is binary fission amoeba different from that of leishmania? Why don't multicellular organisms reproduce by fragmentation? Describe the process of regeneration in planaria and discuss the advantages of vegetative propagation.